right? Great. Yeah, so I'm going to talk to you about, again, bio-inspiration, in particular biomimetics, but for a really interesting class of organisms. And it's not just octopuses, it's all of the cephalopods. It's things like snails, right? Things like octopus trunks and things like my tongue, or the tongues of just about any animal. These are soft manipulators, ones that do not have rigid parts. We just heard a lovely talk from Maria about the articulations of the hand and how it can be used for those many, many functions. Well, cephalopods have solved the same kinds of problems. And I hope to show you evidence of how good they are at dexterous manipulation. And they've done it all without rigid linkages, without bones. It's all done with soft tissue, working against soft tissue. And this is a frontier where I'm happy to be standing, and where we really don't know very much. And a lot of what I'm going to tell you today is just giving you the sense for framing the problem and giving you a sense for the challenges that are ahead. We've made good progress, but the mysteries that are present in the animals are still waiting for us to get to. So some acknowledgements. Jun Wu helped to program the simulations, as did Pradeep Settler. Michael Kuba helped with some of the octopus experiments that I'm going to say. He's now a postdoc in Jerusalem. Apurva Kapadia, Will McMahon were engineers working with me to develop the robot sucker I'll show you. And Faiza Arshad was a technician that helped out substantially with the arm and sucker coordination and manipulation work. Also, I've had really wonderful collaborators in this field. The primary people who work in cephalopods, you probably have seen Roger Hanlon's work on nature shows and so forth. Anytime you see a breathtaking picture of a cephalopod, a cuttlefish or something out in the wild, undoubtedly it was taken either by Roger Hanlon or by Mark Norman. And the better ones, I think, are taken by Roger Hanlon. And Mark Norman does beautiful work too. Bill Keir is a really important fellow in this. You know Bill? Bill Keir? He's the fellow who coined the term the muscular hydrostat, which is the concept that's at the heart of this talk. And he had a lot to do with our work and great help. And then these fellows here are really first-rate engineers. Darren Dawson in controls, Chris Ron and Ian Walker actually knowing how to build these soft manipulators and things. And this work was funded by my university and by DARPA. So my laboratory, as I told you before, is interested in biomimetics. And just to be clear about this, the idea is that I'm really interested in understanding how behavior is controlled and coordinated in organisms. And so we have these feedback loops, you'll remember from my previous talk, where we do animal experiments and robot experiments in parallel, and hopefully circle in to understand whether or not the animal behavior is working the way that we think, that the control systems are working the way that we think. We want to learn how brains work. So at each one of these points, if we fail, we go back and we develop new hypotheses, we do more robot experiments, we do more animal experiments, and then hopefully we converge on an explanation. And as I told you in my previous talk, there is a logical flaw in the idea that if we actually succeed with one of these implementations, we know something. We really only have success when we reject the ideas, because we can soundly reject them. So every time we have a robot that works, we push it until it breaks, right? Or an understanding. And this is the real reason I put the slide up, is that the large arrow here is on understanding how the animals work, rather than in developing new technologies. And so there are lots of shortcuts I could take to move towards technologies, but I've actually tried to tackle the problems of what's going on with the animals. I'm happy to generate new technologies, but the road that I take is a somewhat different one. Okay, and that's what I'll be talking about, a mixture of both. Just wanted to mention a little bit, again, about the way that our laboratory works. We do robot work and simulation work, but we also make it a point to have the same animals under the same roof, so that right there, we can use the animals as a yardstick for performance and collect the data. And we have this wonderful facility, a major cephalopod facility where I am, and so we can keep cuttlefish, octopus, and we even have chambered nautilus, which are an amazing animal that I unfortunately won't have too much time to talk about today. But keeping these animals in good physical condition for experiments is a difficult thing to do, and we do it very well. But the most important thing is that we have the animals and the robots under the same roof. Okay? So here's a video. I'm going to show you a lot of videos to stimulate thinking. Here's a video of our octopus Benji from about 5 years ago. Benji is up against the front glass of the tank. This is not speeded up. This is actual speed. I thought I had this on continuous loop. You'll notice that Benji is moving his suckers in a coordinated way. The suckers are moving, the arms are moving. What you can't see is that we've smeared the glass with something that Benji really likes, the smell of little clams, right? And so what you see here is that there are many appendages here, and the animal is actually coordinating the movement of those appendages in an exploration. And you'd like to imagine that maybe the animal's brain is taking in that information in order to direct the search. The animals have 300 suckers per arms, 8 arms. The sucker rims, as I'll show you in a little bit, are covered with chemosensory and mechanosensory sensors. They are the primary conduit of information about the world to the animal's brain, at least on contact surfaces for tactile work. They have wonderful eyes and wonderful visual systems, but think about coordinating those appendages. Similarly, on the motor side, and apart from the sensory side, these animals are very, very flexible. So this octopus is going to fit its body through that little hole. Okay? Think about the computations involved in reshaping that fluid body to be able to get that animals out of that hole. And they can do smaller holes than that. In fact, they're limited by the only hard part that they have in their body, which is their beak. They have an opposable beak, and they can fit through any space, right? And clearly the animal's solving problems here getting through, but if you root for him, you just know that he's going to get out, right? Think about that. Think about controlling that, and that the brain of this organism does it. This is not a random movement. This is a coordinated, purposive movement that this animal is doing. It wants to get out of that hole. It wants to escape. And to be fair, the animal has evolved to do this, right? This is not a hard problem for the octopus because it evolved to move its way through cracks between rocks, to be able to escape predators and seek for food. It's just that now we've cheated and put it into a glass container so we can see everything that's going on. Everybody suitably impressed? Everybody ready to build one of these? Yeah, that's the thing that we're interested in getting at.
Is that the first time that it actually said that particular box?
Yes, it is, and they don't care. I mean, if you want to go online, there's a wonderful video called Incredible Suckers that I couldn't get before. Yeah, Incredible Suckers. The thing that truly amazed me is I saw an octopus about this big go between 2 plates of glass that were separated by 5 millimeters, okay? Stretched like this. That was the first time the animal ever did that. Now, stretched about this wide, because it's between 2 plates of glass, you can watch what the animal is doing, okay? And you can see the arms shoot out through that plate of glass, the suckers attach, the animal pulls itself through, right? Attach and move, the legs behind, push it. That coordination, for the very first time that the animal had to do it between those 2 plates of glass, it was capable of doing it. Now, its brain was squished flat, its suckers were squished flat, the muscles in its arms were squished flat to 5 millimeters, and yet it was still able to perform the task, okay? I can't do that, okay? Not with my body and not with any of my agents, but this is one of those real challenges for biomimetic systems that we would like to understand. Remember from my last lecture, the octopus is an existence proof that it's possible.
So there is no endoskeleton inside?
Not specifically, not a rigid skeleton. I'm going to talk about what they do have, which are networks of collagen fibers, which flex and provide mechanical support at places where it's needed. But it's very complex, this internal.
So the stiffness, when the stiffness is desired for the task, the stiffness is provided by the muscles?
Exactly. This is one of the main principles of my talk is that the muscles can be dynamically allocated. The same muscles can be dynamically allocated to provide the force when needed or the stiffness when needed on the different parts of the tissue that's required. Okay, so I guess I have everybody's attention. If not, you're dead, right? This is the stuff that really amazes me. So this is a quote from the literature, which I'm a very big fan of Martin Wells. He did wonderful, wonderful work. And I think I've convinced you with this video, octopuses move in mysterious ways. Being flexible, the movements that they make are often difficult to specify and correspondingly difficult to investigate. Literature does not contain a description of octopod walking comparable with that of six-legged gait of insects, or the 10-legged gaits of lobsters, or the stereotyped locomotor patterns of snails or polychaetes. We know how worms move, they're soft-bodied. We know how snails move, they're soft-bodied. But we don't have the same sort of thing for the octopus. Descriptions of posture run at very similar difficulties, perhaps partly because of this. Research into motor control in cephalopods has proved less attractive proposition than research on sensory analysis and learning. Learning is something that we've looked at with octopuses to a tremendous extent, and we're impressed with their cognitive capabilities generally. But the one big thing I need to tell you here is one of the main reasons for this is that we have not been able to access the nervous system in an electrophysiological preparation. The flexibility that these animals have and the lack of a hard outer shell means that when you stick an electrode in, you don't hold a cell for very long. There are muscles inside the brain which move the tissue around when the animal needs it. Squeezing between those two plates of glass, they can move the brain as they need to inside. So we have almost no electrophysiological data, very little central electrophysiological data available. Yeah.
Do they have a peripheral plexus?
I'm going to come to it, and indeed they do. It's an important design feature. Yeah. It's one of those interesting combinations of a distributed and executive nervous system with a completely different architecture. So there's a lot of questions you're going to want to ask me, and I'm going to have to say we don't know. In particular about the control, simply because the data have been so hard to obtain. However, a number of laboratories have really thrown themselves on the sword in the last 10, 20 years to develop these techniques, and we're beginning to get a view into those control systems. And I think it's the verge of a very exciting time. Okay, so another little illustration. Okay, this is very, very close. So here's an octopus, right, with its suckers, and what this animal is going to do in a moment, this is a bit of Reuters footage from the internet. What this animal is going to do in a moment is going to go into a tank, it's going to come up to a glass jar, there it is, and it's going to learn how to be able to lift the lid off of that jar, because there's a yummy piece of food inside. And you can see the animal is doing trial and error things, attaching the suckers, trying different arms. Eventually you'll see the suckers attach, and you've got coordination between the suckers and the arms going on there. He's able to lift the lid and then reach inside. Okay? The central nervous system of this organism can be applied to new tasks, apart from those stereotype tasks that I told you about, being able to go between plates of glass and into things like that. Similarly, this other video is work that Benny Hockner has done. Another unnatural task for the octopus is this striking task. I'll show it to you again. Okay, I've paused it. This octopus has been trained by Benny Hockner and colleagues to strike at this green disc. Now this is a kind of unnatural task for an octopus to do. It has to lift its arms and use its eyes to guide the strike towards that green disc. And as you can see, the animal is able to do it. And there are two threads that are supporting that green disc. And you'll see the animal kind of grabs a hold of them to be able to do it. The animal was rewarded when it did that with a bit of fish. So the point is that it actually can do arm-eye coordination using those appendages that I told you about that have all that incredible flexibility. They've been around for a long time. This is a fossil from 350 million years ago. It's very, very rare that you get a fossil that preserves the soft tissue. This one turned up in 2009. And the interpretation, actually you can kind of see it yourself, is that the arms, the form of the octopus 350 million years ago was essentially the same as that today, with suckers and arms and glia and a whole bunch of other structures. Very, very old. If we look across the different classes of cephalopods, the major ones, right, the squids, the cuttlefish, and the octopuses, as well as the nautilus that I showed you before, they have a common bow plan, an organization to their brain, which is in common across all of them. If I look at an ant brain, or a Drosophila brain, or a lobster brain, I can see that common structure in the different parts of the brain and their arrangement and coordination, connections. If I look at any kind of a mammal brain, I can know my way around it, and even to a certain extent, I can extend that down to fish. The same thing applies to these free-swimming, highly social squid, these close to the bottom, free-swimming and not so social cuttlefish, and the solitary, bottom-dwelling, benthic, manipulating octopuses. Okay? The core brain, going back almost 550 million years, of the nautilus has 13 lobes in it. These all have about 35 or 36 lobes. The 13 lobes that are in here are homologous with the 13 core lobes that are inside these so-called coleoid cephalopods. What's been added are these superstructures here, which are tremendous learning capabilities. But the basics of the motor control is presumably in this basal pedal ganglia.
What's the evidence for early Cambrian?
There's fossil evidence from these guys. They have shells, so they're preserved. And you can look at the points of muscle attachment on the inside and see that the contemporary forms preserve the same sort of muscle attachments, the same muscle groups, and so forth.
That was presumably in the Cambrian, not the pre-Cambrian.
Oh, I see. 505 million years ago.
Okay.
Thank you. Yeah. 505, a transposition of digits. Yeah, 505 million years ago. Thanks. They survived several major extinctions, and those are always bottlenecks for the evolution of innovations. Okay? Some numbers about the organization of the octopus brain. About 500 million neurons in the entire central nervous system, billions more in the peripheral nervous system. 3/5, this gets to your question, Joe, 3/5 of the CNS neurons are in this peripheral sort of ganglia. 3/5.
How were those numbers arrived at?
J.Z. Young had an army of people who looked at slides section by section and did all the statistics to be able to say, okay, we've done this many, many times. They did it by brute force between the 1940s and the 1970s.
Is there any EM?
Any EM? Yes, tons of EM as well. They were very frustrated not to be able to do electrophysiology, so every time they had a question, they refined their histological techniques. There's plenty of EM, yeah, and Grasso did some beautiful stuff as well. So this is the octopus. This is the top view of the octopus nervous system. The section through here is kind of just off the midline here. Huge optic lobes. These are the optic lobes. A lot of visual information processing in these animals. And then these lobes right here that you see on the top are those learning lobes that sit on top. And you'll see them kind of here. You see? Correspondence there. Series of these lobes. Benny Hawker's been doing wonderful learning studies with lesions in them. This is the central brain, which seems to have the representations of motor programs, perhaps, and certainly the body representations and the visceral organization. When people penetrate in here, recent work, Letizia Zollo in Genoa, they have not found any evidence of maps in here. Instead, every single area seems to have motor capabilities, seems to have primary olfactory, tactile, chemosensory capabilities in every single area, and no apparent maps with regular recordings. I said in recent years people have begun to do this. It's a very different type of brain organization. Each arm has a very elaborate nervous system with about 43 million neurons, okay, inside each arm. Those connect through a ring that moves around between the different arms to connect them. That's that brachial ganglia. And that is what communicates with the brain. Okay, so those 3/5 of those neurons are out there in the arms, and they're also in this central pedal ganglia that connects what all of the arms are doing. It's a very different way to organize a brain.
Is there any evidence of microcircuits?
Tons. Microcircuits?
As in repeating microcircuits across the...
Yes, particularly structured around the suckers. And you'll see a little bit of that in a little bit. The anatomical structures of the suckers induce a local specialization analogous to what happens along the length of the spinal cord with those repeated units over and over again. And then microcircuits in the brain, there are characteristic cytoarchitectures that are present in different structures, like in the subfrontal lobe here. There's a characteristic array of orthogonally arranged fibers that repeat. Oh, that's very important. Yeah, there are things that look like, structurally like the cytoarchitectonically, like the cerebellum in terms of their arrangement. Yeah.
It's like parallel fibers.
Lots of parallel fibers and climbing fibers. They don't call them that, but structurally they're very similar. I think I've done my job with this. Yeah, good. I raised some consciousness about that. So I've spent a lot of time thinking about suckers, and we're going to delve down into the suckers. And I want to point this particular thing out about suckers. These are the things we all used as kids, right? The things that you throw at a wall, and then the air gets squeezed out when it makes contact, and then it's difficult to remove, right? And the principle by which that works is that the rubber gets deformed, and it produces a negative pressure space inside, right? And then when the rubber expands back out, you get attachment, right? Everybody follows that, right? You compress it, and then when the rubber relaxes back, it produces a negative pressure. So when you're trying to pull that thing off the wall, what you're doing is you're working against the strength of the air. The air doesn't want to be broken apart, and when it does, it cavitates, and you hear this pop, right? And it comes off the wall, because the attachment is really produced by the material properties of the air or the water. Cool? Very powerful. It's as strong as the bonds between the molecules that hold the air together, and yet it's releasable if you can deform or re-deform the shape of the attachment. Now, the octopus sucker is a much, much more sophisticated structure than this. Squids and cuttlefishes use this principle to thrust out tentacles to be able to attach to a prey item, and they move very, very quickly to produce that attachment, a lot like this dart gun. But octopuses have their suckers hanging off of the arms. And so I'm going to show you in a moment, there are 3 parts to the arm, but I want to just point out these muscles here, the extrinsic muscles. Once an object or a surface is attached, it's attached very, very firmly, and you actually have to break the water, break the material bonds of the water, to cavitate the water to break that seal. Okay? When something is held like that, those extrinsic muscles, like we saw before, the movements of the eyes, you can actually move the suckers around, right? Like a universal joint. And what octopuses will do is they'll actually pass items of food from one sucker to another. By analogy with our recent talk, the suckers are the fingers of the octopus. They do the dexterous manipulation. And the arms are able to grab things by wrapping around them or supplying tremendous force.
You mean that the suckers are adapting according to the task, to the manipulation task, and moving in the space?
Yes. And I'll show some.
Actively moving, not passively.
You said it better than I did. That's what I should have said. They are active agents and not passive agents.
So because this is the difference of fingers.
Yes.
Okay, it's interesting. That's new.
Yeah, I'm not going to, that's right, that's right. And I am not going to push the analogy with fingers too hard. The real analogy I want to say is they're used for fine manipulation. For fine manipulation of objects. And there are reasons for that. Now, last night Joe asked me about these, about the nature of these suckers. So if we look at squid suckers, you see that it has this rim here, and it has all these sharp teeth sticking off. These are actually kind of rigid chitinous teeth. They reflect the function of squid suckers, which are to reach out and attach to something and pull it into the beak, right? The real purpose of this sucker is just to have a one-way attachment, a one-way ride to the mouth of the octopus. How many people saw 20,000 Leagues Under the Sea? Lovely, lovely movie, right? Where the squid attacks and starts pulling people out. They did a pretty good job of depicting the squid tentacle. The squid tentacle is an appendage with suckers just on the end that moves through the water, makes surface, and then can retract to pull the food into the mouth, that one hard part where the chewing takes place. And Joe reminded me last night at dinner that some of these species of squid in the deep sea have nightmarish arrangements. They've given up on suckers altogether, and instead they have many, many sharp spikes, like some kind of a medieval flail that are intended just to go out and hook into whatever it is to pull it in. Now compare that with this beautiful octopus sucker, right? Smooth surface, delicate, not serrated, loaded with sensors, right? We have about 10 to the third primary mechanoreceptors and 10 to the fourth primary chemoreceptors around each one of those sucker rims. And in fact, like the nasal mucosa of humans, this surface is constantly regenerating. When you keep octopuses in the lab, as I do, one of the things you have to keep cleaning out of your filters are images of this skin surface, because every day they shed this sensory surface and regenerate it. So 300 suckers times 8 arms every day. If you have 100 of these octopuses in the lab, you've got an awful lot of tissue regeneration. These things are soft, and there are good reasons for it. So the mechanism of the octopus sucker, here it is in cross-section, is pretty simple. The soft deformable tissue here, this region, is referred to as the infundibulum, okay? And this part right here is devoted functionally to producing a seal. And what this thing does is it can reshape itself to conform to whatever surface is necessary. Once a corrugated surface, complicated surface, it just extends itself down with sensory feedback loops to actually conform to the surface and form a tight seal. When that happens, this interior volume of fluid, just like that sticky dart we threw at the wall, is now isolated. And this portion here, which is called the acetabulum, has a very powerful muscle on it. When this muscle contracts, if it contracts along this way, the entire rest of it is going to lengthen. And so what happens is you're going to be increasing the volume inside of this chamber, right? Producing negative pressure. However, as we said yesterday, Mitra and I, water is incompressible, right? Water doesn't want to have the volume increase, and so what you get is a tremendous amount of force here. In fact, they can generate up to 2 atmospheres of negative pressure relative to the ambient inside one of these suckers. It's been actually measured. Okay? What that means is that you have to break the water, which has a high bulk modulus, in order to break the seal. Okay? So division of function, seal formation by soft adjustment of the infundibulum, attachment by the acetabular cap, and then the extrinsic muscles that I told you about before, allowing whatever's held to be rotated.
How does it contract the cap?
So this is a massive muscle. It's an important question because we have to think very differently about how the muscles work. This is a cross-section on which I based that diagram. And you can see here that there's a massive muscle there. When it contracts, the muscle fibers are aligned this way. So when it contracts this way, this gets shortened. But it isn't like a jointed connection between 2 points of attachment. What's happening is this is a muscle that's contracting and changing the geometry of the entire muscle itself. So when it contracts this way, it's got to expand in the other way. It becomes thinner, and that's what produces the pressure difference. Good? So, as we said, the animal can contract each one of those suckers and produce that negative pressure. But of course, it doesn't actually change shape because the water is incompressible. It's doing a lot of work on the water to be able to produce a really rigid attachment.
What's the approximate size scale?
So this one is probably about a centimeter across. When you get to work with Pacific giant octopuses, as I have, you can get really excited about suckers, right? So you can have one that's this big around, attached to the glass, and you can really do some interesting work with such a large thing you can actually handle and hold. Down the length of the animal's arm, you can get them from being, say an octopus about this big. Next time you have a polpo at dinner, take a close look. You'll see that the suckers get smaller and smaller in proportion as you get to the end, and there's a range of sizes, which will be important in a little bit.
The child suckers, you push the fluid out, so octopus suckers don't do that as well.
I'm sorry?
When a child sucker, like the dart sucker,
Oh, that's right.
you push the air out or the water out.
Right.
But with octopus suckers, they don't do that.
That's right. They maintain and the same thing is really happening with a child's toy sucker. What happens is you push the air out, but there's still some air in there, and that's what makes the attachment. What happens is the mechanical force of the sucker striking the wall, the child sucker as we call it, deforms the plastic. So the momentum pushes it in, and then you're getting that negative pressure inside. So that's the only way you form a seal is if you enclose some fluid. And it works for mercury, it works for oil, it works for water, it works for air, it works for helium. Doesn't matter, it's a property of fluids. The attachment strength is determined by how strongly bonded the molecules of your fluid are together. And water is outstanding for that, because it's an incompressible fluid.
The negative pressure of the fluid is because you're pulling back
Yes.
to create a negative pressure. Does that determine the strength of the contact as well?
Yes, that's an important point of control for the animal. If the animal contracts this muscle a lot, it's going to generate a bigger geometric deformation and more pressure. If it does it a little bit, it'll have a loose grasp. It can actually, in an analog way, control the strength of the grasp. And the thing that blew me away when I learned about this is the limits on the strength of the attachment force of the octopus sucker are set by the properties of the water. They can trivially cavitate the water inside of here, right? They have enough spare muscle power to be able to do it. Yeah, don't worry about it. Everybody knows what cavitation is? Cavitation is when, guys, students, do we know what? I figured you don't, so let me tell you. Cavitation is when you put so much stress on the water that the molecules begin to pull apart. You put it into a cylinder and you extend it and it goes pop. You get a popping sound. That's cavitation. So what's happening is you've reached the point where the material property of the water changes and you have gas being formed basically inside. And when that happens, you have that release on pressure, you induce a state change on the water. Octopus suckers can do that, changing it from water to a gas, from fluid to a gas.
What sort of pressures do you need to get cavitation?
2 atmospheres.
That's quite low.
Yeah, for water in the ocean. And there's a lot to say about that, but no time. Okay, the control of these suckers, and as I told you, there are lots and lots of neurons. You have primary sensory neurons here, mechano and chemo. You have proprioceptors inside of here. There are proprioceptors throughout. And these project up to a sucker ganglion, one that sits over every sucker. And they'll also project up to the brachial ganglion. And the brachial ganglion form this chain in this diagram here. So here we are looking at the arm in cross-section, and here are a chain of those brachial ganglia looking down on them. And they're interconnected, so they communicate with one another. But the sucker ganglia do not. They're freestanding. They have to pass their signals through the brachial ganglia to communicate up and down. And then descending-wise, there are muscles that will innervate the various structures that I told you about with motor neurons from both the brachial ganglia and the sucker ganglia. So this distributed control is something we can look at. We have no electrophysiology to be able to tell us what signals are carried on these wires. All that we know is that the connections are present and information will pass between them. Okay. So we're able to do this sucker contact analysis. When a sucker is attached to the glass, a surface of glass, we can see a visible and optical change in the size. So we have automated systems that allow us to track 300 suckers at a time on an arm and compute whether or not they're attached, stream that data out, and be able to analyze the coordination of the suckers. So we developed this numbering system where we number the suckers from 1 to however many we've looked at, from the base with low numbers and the tip with higher numbers, and then the even and odd numbers, because the octopuses we've studied have an alternating set of rows. You get even odd numbers on the left and even numbers on the right. Okay? So this bizarre looking diagram is looking at contact analysis for a set of suckers. And so the vertical axis here is the individual sucker. So we're looking here at about 25 suckers down the length of the arm. The horizontal axis is time, and the color code tells you whether or not the sucker is attached. So you can actually look at the pattern and the dynamics of all the suckers and how they're coordinated. And when we do this, we can get some sophisticated statistical analyses. We can compute the probability that any given sucker will be attached when its neighbor is, and we get patterns that look like this. Right? Communication between the suckers. Everybody clear on that? If I sort of sum along here, I can compute the probabilities that any given sucker will be attached if its neighbor is. And then this comes to your question, Maria. Oh, those things have gone out. This is actually looking at the dynamics of that. This is one behavior we call the arm probe. And what you can do is you can do like a cross correlation. You can shift all of the suckers into alignment, so each one is a focal sucker, and then you can look and see whether its neighbors are attached when it's attached. So 0 here is the sucker of interest, and the correlation here says that it's always attached when it's attached. All right? And if we go this way and that way, you see that there's this staggered alternation between the even and odd numbers. In this particular behavior, you've got this alternating stepping between adjacent suckers. Okay? And this is a behavior called arm probe, where the animal is like I showed you in the beginning, Benji, with the suckers kind of spinning around. You can actually, remember the Benji on the glass moving those suckers? Well, that's what you get. You get this alternation in the neural control between them. This is a different behavior.
Or is that between so they're in two columns like that in that picture?
Yeah, so the sequence is 1, 2, 3, 4. And so if I were going from this sucker to this sucker, I'd be going from 0 to 1. And if I were going from this sucker to that sucker, I'd be going from 0 to -1. I've just kind of shifted everything into a sucker-centered coordinate to accumulate the statistics for all the different neighborhoods.
You've thrown away edge effects, right?
Right. Yeah. So what you can see here, and you don't have to follow the details, but this is a different task. When the animal reaches its arm up, attaches to the glass, and then shortens its arm to pull itself up, that's different from this arm probe. And what you can see here are these sequence of blue lines. They're in there too because the pattern was constant. But this is early in the arm reach, and then you see it narrows down to the final state of the red there with that alternation. So what's happening here is a selection process of a particular neighborhood that's active. And so the patterns of coordination of the suckers, in fact, vary with the task that the animal is doing. The attachment of the suckers vary. Okay? I've accomplished my mission, right? Everybody's beginning to be impressed with the complexity of the computations and the integration of all those complicated mechanical properties with a neural controller. Right? Questions, everybody? I know it's kind of an abstract figure, but the take-home message is the patterns of coordination vary with the task for attachment.
It has a tendency for either left suckers to be on or right suckers.
At the same time. And remember, I've shifted the left and the right on top of one another, so they're kind of mixed together. It's more like the alternation is present there. And you could view that as a form of lateral inhibition, right? Between the different ganglia that I told you about. So we built this apparatus to test the kinds of coordination that the animals did. We built a transparent dome, and I think the colors in this have washed out, unfortunately. It's not as clear to see, but glass dome that the octopus can see a crab under. Glass bottom tank, so we can actually film the behavior of the octopus and the crab and the dome. And electromagnets, which allow us to vary the strength of the attachment. So the octopus approaches, it wants to lift the dome to be able to get at the crab, and we make the task harder by increasing the strength of the electromagnet, and then look and see how the animal uses its arms and its suckers. Right? Simple enough, and I can go into more detail, but just to show you the results, this is a wonderful video to watch, okay? This is one of those examples, okay? So we put the animal into a chamber. We're looking from the bottom up through the tank right now, where it can get its arm in here. Only one arm at a time to do the task, not multiple ones. If you give the animal freedom, it goes up, lands all of its suckers and all of its body on top, lifts the whole thing up. But here we force the animal to do the task this way. And over here, you can see on the side, here's the arm coming out, there's the dome it's mounted on, and these two here are two magnets. This is before we had the electromagnets, but it was a beautiful octopus. So if you watch, you can watch the unfolding on both sides, right? The animal actually gets maybe a sense for the geometry of the object, feeling its way around it. And then it lays the arm across, and you can see when the suckers attach, you can see the increase. That was an interesting thing, the increase in the diameter telling you where they're attached, and then the animal continues to probe with its suckers, trying to find good purchase, and then it lifts the dome. Let's watch it again, and I'll point some other things out. I know it's always hard to view these things. The animal is going to explore the surface. Remember all those sensors on the sucker surface, right? Tactile exploration, getting maybe a feel for the shape that its eyes couldn't get. Now this is interesting. You see those points of attachment there? They attach, and you see he's moved the dome this way. Right? Maybe moving it into a position where he could get a better grip on it. Now the suckers are attaching here, and the suckers attached there, and what happened is the arm lengthened. So there were suckers attached on the two sides, suckers on the dome. The arm lengthened in between those 3 points of attachment to lift the dome and break the seal. He was able to use all the strength of his muscle to lift it that way, reshaping the arm.
Was it also attaching to the actual floor of the, it seemed to be attached to the floor around the
Yes.
push off?
But the analysis that we do allows us to, and this is a bit more laborious than the automatic tracking, allows us to describe in that same way the pattern of attachment. So early in that sequence, this is the sequence we just looked at, you see this kind of random pattern of connections, right? And here is where it moved the chamber. And you can see this pattern where it had some suckers that are attached to the chamber itself, to the floor of the tank, and to the floor of the tank, as you saw visually. And then when it pushed it, right, those suckers were attached. And the push was achieved by lengthening the arm, if you remember. Does the same sort of strategy here when it comes to lift. Here's the point in time where the magnets fall off. Right? And I mentioned this is exactly the same configuration as before, suckers and time. So here's the point where you have this kind of bridge being formed with attached suckers, and the lengthening is really happening where these unattached suckers are. So the animal can reshape its arm. And I'll tell you about how that works in a moment. Okay? So,
Is it under visual control, or is this behavior?
I don't know the answer to that, because this was a hard enough thing to set up and to get the animal to do, and so we didn't blindfold the animals. To motivate the animal to know that the crab was there, we let it see through the plexiglass. But I suspect that there's probably a lot of tactile activity there because of that original exploration. If it really had a sort of visual representation of the task, it should have just put the arm down and done it. Instead, it was feeling and exploring initially.
What prevents him from coming through that hole?
The sheer force of the materials we built it out of. Yeah.
It's big enough.
No, that was part of the reason I like this is that was a monster octopus. It was a big octopus, a very big octopus. So the diameter is there, and the animal might have gone through, might have tried, but it didn't. It didn't try. But also, we drilled that hole to be small relative to the size of the animal. They don't do, you know this, Joe, yeah? You don't work with children and animals. They don't do what you expect whenever. Here we got very lucky. And just a quick mention, this is work that I do with my student Stavros Hadjisalum, who I just realized I forgot to acknowledge. Same kind of, more complicated sorts of plots. Cross correlations of the movements of individual suckers. So when one sucker moves, if it's concordant with the others, you get a positive correlation, and if it's in opposition, an anti-correlation, you get a negative correlation. And what you can see is that those extrinsic muscles that I told you about that move, that make the finger-like movements possible, are what are going to produce the sucker movements that we've observed here. We have a complicated method we've used to pull the arm-independent sucker movements out. Okay, so these are descriptions of those. And what you can see is this is a delay in time from a particular event, looking at the coordination between the suckers. And this is the correlation between one sucker and all of the suckers that are farther down the arm from it. Okay? So I'm looking at this sucker, and how does it correlate with the next, and the next, and the next, and the next? This sucker, and the next, and the next, and the next are each line. And then this is the amount of agreement between them.
Agreement in what? In whether they're contracting?
No,
or
movement.
What is movement?
The sucker rotates.
So it's a rotational velocity?
It's whether or not the sucker moves at all. So if this sucker moves and the other sucker moves and they agree,
What,
if one of them doesn't move and one of them does move, then it's a negative correlation across very long time series.
so it's like a binary measure of velocity. If it's moving at all,
Yeah.
you give it a 1 in any direction. So you could get a 1 even if they're moving in opposite directions.
That's right. And we've done the more advanced analysis, but the graphs are even harder to understand.
Yeah.
So the point here that I want you to take is red means correlation, positive correlation at the 0.01 level. Blue means not at that level. And red means, black means a negative correlation at the 0.01 level. And what you can see is that depending on the task, the animals move the suckers in a coordinated fashion that's different for each task. Can everybody follow that idea? Right? So we gave these animals different tasks. This is the arm walk where the alternation is happening in those slides that you saw before. The movement is correlated, you expect it. This is the arm carry. This is where the arm is kind of dragging along behind the moving octopus. And there's sucker movements in there. And this is the arm probe where the animal is reaching around trying to explore a surface. Okay? So we have attachment that varies with the task. We have responses to the amount of force that the arm needs to produce in order to deal with the attachment, and the movements of the suckers all vary with task. So the arm, I hope you're now even more convinced, is this coordinated structure for producing manipulation at fine and large scales. So how is this achieved without rigid parts? And this is really the muscular hydrostat stuff that I was telling you about. How do we think about a soft system to be able to do all of this? Well, Bill Keir came up with the concept of the muscular hydrostat. And I'll just point out that there are plenty of examples of structures that are driven by muscular hydrostats, which I know I haven't defined yet. Tongues, right, in bats, lizards, cephalopods we've talked about. Elephant trunks, which are capable of lifting very great loads, operate on this principle. No hard bones, the muscular hydrostats, cat's tongues, and so forth. And the principle is very simple. If you have a massive muscle, you can think of it as containing a volume of water. There are hydraulic processes where water moves from one region to another, and the net displacement of water produces an inflation or deflation. Starfish tube feet are an example of that, right? Where there's a net movement of fluid. That's not what happens with a muscular hydrostat. The volume is constant. And so if I have an object, which a cylinder, say, and I contract along the length, it has to expand along the diameter, and so it increases in size. If I have millions of these side by side, and I can control them individually, it becomes possible to reshape the tissue as well as adjust its stiffness by activating those patches of muscle. And I'm going to show you a lot more about that. But the principle of constant volume is the important one here. No attachment between two rigid points, rather a reshaping of the tissue. Contraction along one dimension produces expansion along the others, at least one other. As it turns out, the structure of these systems is built so that there are fibers which typically rationalize the problem and make it so that the expansion in the other dimension is limited to one other dimension. So there are 3 dimensions of space. Contract along one, rigid fibers keep it from expanding along the second one, so it's forced to expand along the third. Good? Okay, so here is I'm going to show you a video of the Octom robot that I was happy to work on with Ian Walker, Chris Wren, and Darren Dawson. They were actuated, since we don't really have real muscular hydrostats, with McKibben actuators. And McKibben actuators do this net flow. You can see that as you open a valve here, you can put more air into here or increase the pressure on the inside. And so it'll expand in this dimension and contract in the other. You see how those parts are contracting? And this is a cartoon version. This isn't the actual McKibben actuators that they used. But what you get from this is impressive in terms of a capability, a soft arm. And I should say that this arm that I'm going to show you was made of 9 of these McKibben actuators, really rather long. So there were 3 sections with 3 McKibben actuators on them, right? And so the 3 were arranged in parallel, and if one expands, what's going to happen to the other 2 if they're not activated? Well, that expansion is going to cause a bend, right? If 2 of them expand, it'll cause a bend around the one that doesn't expand. If all 3 of them expand, you've got an increase in stiffness in the entire structure as well as an overall shortening. So there were 3 of these segments down the length of the arm, and you will see it doing various interesting things. So there it is, right? It's got you can't see the McKibben actuators. It's got an outer skin, which is very important. It holds things together. It can form these spirals, and it can lift, soft objects, rigid objects. And the nice thing about it is that if you were to make this very long, instead of being limited in your grasp to just, and it also holds on to things in the face of agitation, instead of being able to just grasp something this size, well, that's fine. The hand is designed to manipulate objects of that size. But if I wanted to lift a trash can, I really can't do that. But the wrapping process is the same for all of them. And, it does all kinds of things. These are demonstrations, right? Everybody got the idea that it's possible to take that general principle and build something which will make smooth, continuous curves. Now, the folks in Pisa right now have taken these ideas and gone even farther with this. Some really neat stuff in terms of flexibility. You see the control was a little bit jerky there. It's because those pneumatic actuators, those are air inflation things. They come on hard and they go off, and there's not very much you can do about smoothing out that control if you want to have the kind of force that's necessary. It's also not what octopuses really do. It's only part of it because there was a net flow, there was a pneumatic net flow of fluid on the inside, right? Inside of those actuators. The real octopus does it by having a force-generating element inside which contracts muscle. Okay. How am I doing for time, Tony?
10 minutes.
10 minutes left? Okay, so I got 90 more slides. I'll go a little more quickly. The architecture of this is a cross-section through a squid tentacle. The architecture is really interesting. Okay, that muscular hydrostat principle I told you is neat, but if you look at this section here shows the muscle fibers in I think, let me get it straight. Yeah, it shows the muscle fibers in red, and then there's connective tissue materials in blue. And so there are transverse fibers that run through the length of the tissue, and when they contract, they shorten the entire tentacle, but simultaneously make it fatter. In an antagonistic fashion, there are the longitudinal muscles run the length. The transverse run at various angles, and when they're coordinated, you can contract the diameter of the entire structure and expand and lengthen it, okay? And then there are these circular muscles that run around the outside that can contract and provide local stiffness as needed and also affect the lengthening, right? And these things can be actuated in opposition. So the other thing that's neat about this is this connective tissue, and that's the other side that's really important in controlling these things. Because if you just have something like an amoeba that could reshape itself, it would be infinitely useful, but it would be difficult to interact with the world. And so these fibers here guide, structure, and channel the direction of the forces that the muscular hydrostats produce in directions that are biologically and application appropriate. So for the animal, if the animal's going to do something, it'll shape its structures that way. So we developed this modeling system which allowed us to be able to basically implement the muscular hydrostat system. Completely from first principles, we set up a space where we could have cylinders or cubes or trapezoids that would contract in one dimension, expand in another dimension. And we modeled it with the pressure from the actual physical forces inside being modeled. So we contract along this dimension, that generates an increase in pressure in the incompressible water that's modeled inside. And there's a resistance, a tissue resistance to that change that resists it, resists that expansion. And so we can actually produce the real dynamics with these individual, we call them MHUs. Okay? So the idea is use the force generation that matches the capability of muscle, apply it to a volume that has the density and properties of water, and then give it the elastic properties of the tissue and play out how the geometry of an individual unit changes. And this never happens in real biological systems, but this is an output from our simulation where you can have a change in geometry that's that dramatic. You can contract along one dimension and produce a very fat cylinder, right? And that's the muscular hydrostat principle. Okay, and here's a little demonstration of it in action. First, we rotate the thing so you can see it's in 3 dimensions. And now we have 2 of these things connected one to the other. There's an expansion along this dimension leading to a contraction, and it's pulling the other one along with it. So the idea is you get that reshaping, and by the way, this is biologically implausible. This was an early test, right? You can actually produce a reshaping of the whole object. Okay? When we build these systems in simulation, we have those MHUs that I told you. We attach nodes to them. These are points where points of force can be applied and transmitted between muscular hydrostats. We connect them with springs at appropriate points that we can adjust the tension on or the stiffness of in order to match the biological materials of the system that we're modeling. And then we have these these unusual things called anchor points, which allow us to fix the actual structure in space. Okay? And the next slide will make that a lot clearer. Oops. I'll go back to it. All right, maybe I'll go in the order I planned. This just shows that we get realistic dynamics out of that. So if we give a command signal to one of these MHUs, this horizontal line, say, it tells it to be active, and we can grade it so that its length changes. We contract along the radius, and you can get these kinds of responses out, and you can actually grade it to do a small change or a very large change in its geometry, each individual one. Okay? And the thing is that these dynamics that emerged from the constants that we put in, the stiffness of the muscle, the force generation capability of the muscles, have rise times and fall times in seconds that are biologically plausible. So we're building this thing from the ground up to have biological dynamics. Okay? And then the skin, here's a little video that shows the skin. Again, we rotate it in space so you can see it. So there's a set of nodes with some springs between them, and we can adjust, and this is a principle, we can adjust the stiffness in some of the springs to produce some kind of movement. Now this is not something we do. For us, the connective tissue is a fixed passive property, but this is a basis of some other models of, muscular hydrostat systems, which doesn't capture the biological flavor of the muscular hydrostat property. And here we're actually applying a sinusoidal force just at the first set of nodes here, and you can see the waves propagating down the length of the structure. So we have this connection structure, and the connections are being restored. That's the outer skin. So these 2 components that need to be in there are the muscular hydrostats. And this is an assembly of 27 of those muscular hydrostat units here in 3 rows. Okay? And when we activate those units, now we have this graphical thing that makes them turn red. And I think this will convince you that we've got some very realistic dynamics coming out. So what we've done is we've just told every one of those muscular hydrostats to contract at its maximum rate. And you can see that the thing moves through space, generates a bend, and what you missed there is that then that system will actually, when the energy has been dissipated through friction, right, the thing will actually go back down to its ground state with the tissues pulling it back into place. The connective springs pulling it back into its original geometry. So the idea is that you have a whole bunch of these muscular hydrostat units that you assemble into space, a geometric arrangement that matches the system that you want to model, the octopus arm, the octopus sucker, the tentacle. You produce an appropriate skin drawn from those literature values. You connect it all together, you let it relax to its ground state, and then you perturb it with, say, an artificial neural network, which can then produce driving signals to that plant. You can tell it, as I showed you the analog signals, you can tell it every one of those contracts maximally or whatever value you want, and you can produce changes. So I hope you see that we have a tool here that allows us to explore muscular hydrostat dynamics and in particular the control of muscular hydrostats in assemblies like that. I'll skip this. This is the control diagram. So here's a robot sucker, an octopus sucker that we built to scale. It has the infundibulum, the acetabulum, and the acetabular cap, and the extrinsic muscles, right? So we're able to build this by taking those functional units and giving them the appropriate properties and connectivity. So 6 suckers in a ring for the infundibulum. I didn't talk to you about the sphincter. It turns out to be very important in the control because that's the fulcrum between the acetabulum, which produces the pressure change, and the infundibulum, which produces the seal. Right. Basically, a lot of these muscular hydrostats need to be coordinated. We anchored it to the fixed point in space, and here you'll see one of these suckers interacting with the world. So there's a sucker, there's the activation, and we deliberately placed it off center so when it makes mechanical contact with this object that it's going to attach to, it actually acts on it, and then there's this rigid one that's the sort of tabletop that it's sitting on. We can get seal formation with this. Right. You'll see it'll actually rotate the object and force it into forming a seal. And then, this is a very interesting kind of experiment that we can do, okay? This is not going to look pretty, but this is what we call the whole 9 yards, where we would actually attach to I think this was a 9-kilogram object. We made an artificial sucker of size 20 centimeters because we were supposed to be building something that would actually work in the world. Important point here is that the simulator that we have here becomes a prototype development software for designing the muscular hydrostats, right? Systems like this. So here, we use biological materials, muscle force actuation, and connective tissue. Here, we used artificial, electroactive polymer force generation parameters, and rubber as the connective tissue. And here, we were modeling it as if it was in water, with the bulk modulus of water, and here we're modeling it in air. So when I play the video, all 4 of these are going to go through the full control cycle of attachment and lifting the object, and only this one is going to succeed because we tuned the parameters to make that work. And you'll actually see how hard these things are to control as well. So there it goes, it's lifting it, and the other ones are going through some unstable oscillations, right? So it actually formed the seal, was able to lift the object, and we did this by modeling the interior volume so that we knew what the compression was going to be, okay? So now, all of this so far has been done with handwritten control. We actually would like to have controls, systems like artificial neural networks, that could control these. I'm going to talk a little bit about that because I have -1 minute, right? Okay. We built an actual sucker that we mounted on the octarm, and if anybody's interested, I will show you later. But I was very pleased that it reached the theoretical limit and was capable of lifting a 12-kilogram mass. It was what the air said it should be able to do. Squid tentacle extension. This is a very important thing that we've modeled because it allows us to get biological validation. So again, it's a pretty simple thing to do. If you have a series of these muscular hydrostats in a row, if you contract them along this dimension, you get expansion along that dimension, and you can produce tentacle extension, right? And okay? That's actually very good. This has been slowed down, but we were able to reproduce the biological trajectory and time constants. So we get 70% strain in 50 milliseconds, which is what you get with a squid tentacle strike. If we use artificial materials, this is funny, so watch closely, right? The force generating capability of muscular hydrostats is much lower than that of EAP. With exactly the same system, see? Watch it again. There it is retracting back. Okay? Birds are really good at flying. They're more maneuverable by our planes, but they can carry heavier payload. Our planes can carry heavier payloads and go farther. We can do much better if we use EAP here. Stepping out a little bit, you can see a little more perspective and slowed down that you can actually produce something that can reach out and strike quite easily. Lot to say there, but basically the take-home message is that we were able to reproduce the biological data reasonably well. I want to say one more thing about this before I go, but we get good reproduction of the dynamics. So this is the strain and this is time, and this curve here shows what happens when you build it out of artificial materials. You get greater strains faster. And then, I'll skip that. We built a recurrent neural network that learned how to control the tentacle. Very hard to do, in fact. But one thing recurrent networks can do is learn sequences. And when we did that, the difference between the biological data and the simulation data went away. That is, we could train it to reproduce the correct pattern. And basically what I think this network was learning were the material properties of the arm that a simple, straightforward, handcrafted calculation could not do. Okay, well, I won't say anything about those interesting points. I will point this out. That's a human tongue, which is a muscular hydrostat, and you can see how it's reshaping like the octopus arm. This is basically going through the different letters of the alphabet in cross-section and fMRI. If you think that we can't control these things with precision, think again. I am speaking to you with a muscular hydrostat right now. The reshaping of my tongue is articulating and reshaping the air that's being pushed out of my throat. It gives you that idea of applications for these things. Obviously, those guys are lots of fun. I just love this guy. And he doesn't want to come, so that's fine. Thank you very much. Sure, they're everywhere, right? Recurrent networks are ones that have loops between the layers. I've got millions of them in my brain.
Where?
My retina, my lateral geniculate, visual cortex.
Of the Hopfield and Tank cell?
No. Well, Hopfield and Tank are recurrent, but the understanding of the retina.
Backpropagation. I mean, to my knowledge, they don't exist.
Backprop is a learning mechanism which has some problems in implementation. But recurrent networks are ones that have feedback loops. You've got them in your networks too, where one layer provides information back to a previous layer. That's in contrast to a feed-forward network. So if you have any recurrent network can be simulated by a feed-forward network if you keep adding layers. The nice thing about this is you can have one layer that learns the dynamics for you. And that's why I did that, is that these things can learn sequences very, very well. You store them in the connection weights. So each one of the expansions down the length of that tentacle had to happen with a particular kind of timing to allow for all kinds of interesting things that were happening there. So if you activated them all at once, you'd get a tentacle extension because there was no way, but you would get weird dynamics and some vibrations. But if you allow, like building in there this temporal sequence by having the units inhibit one another and feed back, you can get a much smoother and faster and more accurate extension by training the thing to do it.
I mean, the loops that I know of are sort of like between the lateral giant neurons and the abdomen, but those are not ones that...
But that's recurrent. Because the information, yeah, it's a loop.
It's not showing backpropagation.
But backpropagation is a learning mechanism.
Yeah.
And recurrence is a connectivity pattern. So when it loops back, when the information reoccurs at the same node, that's a recurrent net.
Any lateral connections is recurrent.
Lateral inhibition is an inherently recurrent process.
Mitra?
We'll talk later.
As the octopus goes down to different depths, the pressure changes a lot.
Yeah.
I was wondering if you look at how the pressure changes influence the ability of the octopus to...
Yeah, let me be clear. I didn't do that work. I was citing the literature, and that was Kieran Smith. And if you want to read some lovely science, you should definitely read Andrew Smith's treatment of this. He took limpets and he took octopuses and he looked at the attachments at different depths. He went out to Big Sur and attached a strain gauge to a limpet in the surf to look at the resistance. Anyway, I think the question you're asking is how do they compensate? And they do. And part of that is that their muscles so overpower the water that they have plenty of residue to spare, and in fact their attachment gets greater at greater depths. The actual literal tenacity, that's the measurement of force that they use. Read the paper, it's lovely. Yeah.
Well, first of all, thank you for the talk. You definitely turned me into an octopus fan.
Mission accomplished.
To make a very stupid example, if you ask me to squeeze some amount of strawberry flavored jelly through a little crack, I can do it with exactly 1 degree of freedom in my action, just push until everything flows through. And they argue, well, the remaining infinitely many degrees of freedom are within the material as such. But because
I hadn't thought of that. This is lovely. Go ahead.
but I mean, you said there are basically infinitely many degrees of freedom
Yes.
in this arm, but does it still make sense to talk about degrees of freedom then? Because this would involve talking about computation, and you just can't compute for infinitely many degrees of freedom.
Yeah. Yeah. So when people talk about these systems, they call them hyper-redundant systems. And people model these also as continua. They make continuum models, which are very interesting. The problem is that you can't figure out the points of neural control with those continuum models, because you can get the reshaping as you want, but if you really want to understand the connectivity that this neuron is affecting this muscle mass and what does it mean for the actual implementation, you can't get there with a continuum model. So that's why I went this way rather than going with a continuum model. And there's a price to pay on this side too. People talk about the whole arm as having infinite degrees of freedom, and I think that's not, strictly speaking, correct, because there's a limit to the curvature that the arm can produce. But it is definitely hyper-redundant. And the nice thing about an octopus arm is that it can produce pretty much as many joints in as many directions as it wants spontaneously down the length of the arm. So to your comment, I guess I would say that the degrees of freedom are immense, but really not infinite. And a lot of people who think about this think about ways of reducing the dimensionality of the degrees of freedom through control mechanisms that might be stereotyped, that animals might learn. And I think there's a lot to that, and it might be a reason why the octopus has such tremendous learning capabilities, is that if it needs to learn how to unscrew a jar, it doesn't just deal with stereotyped behaviors, but maybe repertoires of them that it knits together as it needs them. Thanks for the jelly analogy. I really like it. Yeah.
So a couple more questions. One here, one here, then we'll stop.
Okay, so I saw from the simulations, they were very nice.
I couldn't hear something.
The simulations were really nice, but how far are you, technically speaking, from what we saw in the simulations to a real, actual kind of, let's say, octopus-like robotic hand?
Yeah. I wouldn't build a hand, but a manipulator of some kind.
Yes.
I think that we could do this in a pretty straightforward way with the kinds of artificial muscle materials that we have right now, encased inside of rubber that we could knit together with some material pretty easily. But it will take some time and some experimentation to develop those things and make them work. I think shape memory alloys have enough force to be able to do this kind of thing. Electroactive polymers have sufficient force and force density. One of the things I didn't mention is that the octopus arm, contraction of 23% along one dimension produces 70% extension along the other. And so you have tremendous mechanical advantage. Small extensions of a muscle can be amplified by the geometric arrangement of these items. And so how far am I? If I had the money and the time, I could be doing it tomorrow and begin exploring it, and that would be great fun. And the neat thing that we did that was within reach was to build this prototyping system which allows us to design and work out the issues of dynamics before we actually build something. But we definitely designed this with the idea of building things.
Last question.
I have a question about in the simulations you had 3 colors of these idle sets.
3 colors?
3 colors.
Oh yes, yeah.
And I think you show a cross section of a tentacle of
Oh yeah.
and are there
No. I think the continuum idea is an important one too. So when I showed you that, there are muscle fibers at many orientations, and really we don't know how they interact and slide over one another. The way that I rationalize that is that you're activating all of the muscles of a particular orientation to produce the effect you want in the entire muscular hydrostat. So I was grouping them. Into these units that were functional units that would be active at the time. And so I wanted to have the 3 columns as a way of being able to produce a minimal amount of opposition. And that was not a model of anything real and biological so much as an exercise to make sure we were doing things correctly with the system. But we call that the GM, the general manipulator, because you could reach out and whack whatever you wanted with it, right?
But it would make sense also to have the 3 columns
It does.
in the real.
And I think that the animal can do that and does do that when it needs to by dynamically allocating subsets of muscles that fulfill those roles. And that's what this modeling system does is it says, okay, in a sense, the ABSEMS modeling system that I put up here is impoverished compared to the actual system. That's why I mentioned the sliding of the fibers across, because there will be perpendicular fibers that are present in the real system that can be activated that this thing wouldn't be able to model because we're committed to doing a particular motion. And I'll say about the sucker, the reason we really did this is sucker has so many different directions of action that we needed this kind of system. We couldn't model it with a continuum, and so it grew out of the work for the sucker. And the aim wasn't really to produce something like an arm, which you might be able to do more profitably with a continuum type of model, because then you could get what you're talking about.
Frank, do any animals that live in dry environments use things like suckers? Is it a constraint you have to have some moisture?
We have looked hard to try to find some.
Yeah.
And you find these fish in Hawaii that climb up waterfalls, right? And they're outside of the water and they're on the rock, but they're using water there and they're an aquatic species. The stuff that we did, the video that I didn't get to show you, we built an in-air sucker, and it worked just fine to be able to lift and hold, exactly what it should. I think the big difference is the bulk modulus of water is so high compared with air that you can hold much less effectively with an in-air sucker with a closed volume.
You just can't get such a tight seal? Or?
The seal is not the issue, it's the amount of force you can apply to hold on to something.
Right.
A seal is not a problem to achieve. Mechanical pressure enough, you can do it. Conformal surfaces, you can do it. The issue is how much can you bear, because before your fluid breaks,
Right.
cavitates.
Okay, thank you very much. So we have a short break and then we'll